Hi and welcome to 2.2 all cells specification, just in case you want to pause and read. And we're going to start today with the cell cycle. This here is a diagram of the cell cycle. It's worth noting that this here is the beginning of the cell cycle. So this, the beginning of G phase 1, or G1 phase, sorry, is the beginning of the cell cycle. That's where it starts. Now you can see also that we've got one phase, a second phase, and a third phase. These three phases make up interphase. An interphase is all of the time that a cell spends not in mitosis. So this section here is mitosis, which you should have come across in GCSE. These three phases are interphase. Now within the gap phase 1 or G1 phase, you would expect the cell to grow. So if you imagine the cell has kind of just been produced at this point, so it's going to grow. It's going to produce new organelles, and it's also going to undergo lots of protein synthesis. As you go through the A level, you'll realize the importance of proteins in cells, and the fact that protein synthesis occurs at this stage will become more clear. We then move on to the second phase, which is the S phase. Now the S in the S phase stands for synthesis, and we're synthesizing DNA. So DNA replication occurs at this point. So DNA is synthesized. However, it's worth noting that the chromosome number is maintained. So human body cells will always have 46 chromosomes. When you come to study meiosis, or maybe previously at GCSE, you'll find out that the cell, the chromosome number halves during meiosis. The chromosome number during mitosis is maintained. So 46 chromosomes at the beginning, 46 chromosomes in a cell at the end. We then move on to gap phase 2. This is very similar to gap phase 1. So we've got cell growth again. So the cell is now getting ready to divide. So this was the start of our cell's life cycle. It's going to come to the end of the life cycle here. New organelles are made because this cell is preparing now to split into two. So therefore we want two cells, and both of those cells need to have an adequate number of organelles. And protein synthesis, particularly the synthesis of any proteins that are involved in the division process or the mitosis process. Then interphase ends and we get to mitosis here. You can see that there's 4 phases of mitosis. You've got prophase, metaphase, anaphase, and telophase. You need to learn these and you need to know them in order, and we'll look at them in more detail on the next slide. You've then got cytokinesis, which is the splitting of the cytoplasm. So the cytoplasm has to split in order for the one cell to break into 2 cells. And cytokinesis is the end of the cell cycle. It might be worth remembering from GCSE as well that mitosis produces genetically identical cells, and it's needed for growth and repair. This is sometimes a 2-mark question at A level. What's the purpose of mitosis? The purpose is for growth and also for repair. So if you cut yourself, your skin can repair via mitosis. Okay, the phases of mitosis. Prophase is the first phase. Now in prophase, chromosomes condense, and that means that they get shorter and fatter, and it means that we can visualize them and see them with a microscope. We can't see them with a microscope, individual chromosomes with a microscope, not in prophase. Now also during prophase, the nuclear envelope breaks down. So the nuclear envelope is this that surrounds the nucleus. So that breaks down and that's going to disappear. In metaphase, you can see that the nuclear envelope has disappeared. You've got chromosomes lining up along the equator of the cell. Now, the equator of the cell is the middle of the cell, kind of like the equator of the Earth is the middle of the Earth. You've also got spindle fibers forming and attaching to centromeres. So these blue lines here represent the spindle fibers, and the spindle fibers attach to the centrioles here at the poles of the cell, and they also attach to the centromeres in the middle of the chromosomes. In anaphase, those spindle fibers start to contract and shorten. Now, when they contract and shorten, they're going to pull the chromatids to opposite poles of the cell. You can see here that the chromosomes were X-shaped and made up of 2 chromatids. Here, the chromatids are being pulled half to this side and half to this side, to opposite poles. So we call these opposite poles. You've got the equator and 2 poles of the cell. And then finally, telophase is when the chromosomes reach the opposite poles. You can see that the nuclear envelopes are starting to reform and your cytoplasm starts to split. So the cytoplasm will start to kind of cleave and split down the middle in order to form 2 new cells. Cancer, the definition of cancer is the rapid and uncontrolled division of cells. Now this links to mitosis because often when a tumour forms, there has been some sort of disruption to mitosis, to one of the phases of mitosis. Cancer is usually the result of a gene mutation, and the result is generally that a tumour will form. Now, if we disrupt some of the stages of the cell cycle, we can try and treat the cancer. So lots of chemotherapy drugs aim to disrupt a specific cycle, a specific phase of the cell cycle. Sometimes we can affect the synthesis of the enzymes that you need for DNA synthesis in S phase. So if we disrupt the S phase of the cell cycle, it means that mitosis can't occur. And if mitosis can't occur, then the cell can't divide, and therefore this rapid uncontrolled cell division can't occur. And some treatments can help cells cease as a result of recognition of DNA damage. So usually your body is checking all of the time for cells that have damaged DNA, and some treatments can help the body to recognize these cells and can kill the cells.